Greetings everyone. So today we are going to discuss about the specificity of HIV. We know that HIV is the virus which infects our immune system, and particularly the major valuable asset of our immune system, which is a T helper cell. Now, here as the virus infects the T helper cells, we need to have a concern about. Basically, it majorly infects the T helper cells, but it can also infect other CD4 cells, which means macrophages, dendritic cells are also infected by HIV. So here the HIV virus is highly specific, highly specific to the T helper cells and other CD4 cells. Why? What is the reason? Why does it not infect other types of cells? This is because CD4 cells, such as T helper cells, have CD4 receptors. They have CD4 receptors, and these CD4 receptors are basically uh, the CD4 receptors are the receptor sites where the glycoprotein spikes of the HIV bind attach together. GP120 attaches together and forms mutual affinity with each other. It forms a mutual affinity with glycoprotein spike, as a result of which there is mutual affinity, due to which attachment is possible. Favorability of HIV to attack that particular cell is possible. And after that, CD4 receptors and co-receptors of CD4, such as CCR5, which is found in macrophages, and C into CR4, which is found in T helper cells. Now, these basically act as a site. They provide the favorability, provide mutual affinity for the HIV glycoprotein spike GP120 to attach with these receptors and come in contact with the co-receptors, such that this causes the formation of a particular complex, due to which attachment is possible, and then fusion and other steps are also possible for the life cycle of HIV. Now, here, why does the HIV particularly infects the T helper cells? Because there are other types of CD4 cells as well, such as macrophages, dendritic cells, B cells. uh, B cells are basically these are antigen presenting cells, but why does it not infect it? So the reason behind it is that here, T helper cells is having maximum concentration of T helper cells are having maximum concentration of CD4 receptors, as a result of which they mostly and majorly attack over T helper cells. One more thing, that specificity here is also on the basis of its co-receptors. Co-receptors also play a huge role, such that um, if we are supposed to inhibit the expression of a particular co-receptor, let's suppose CCR5. CCR5 is a co-receptor which can be um, inhibited. Its expression can be inhibited. So if it is inhibited, then HIV cannot infect macrophages, because co-receptors are also involved in the process of life cycle, in helping the fusion process of HIV with the host cell's lipid bilayer. Now, likewise, when it comes to C into CR4, if we are not supposed to or cannot inhibit the expression of C into CR4 co-receptor, as a result of which, whenever uh, HIV attacks over the T helper cells, the T helper cells you know gets infected, as well as there are no particular medicines through which we can artificially inhibit the expression of C to CR4, as we are supposed to do with the CCR5. So in this case, this is we need to have a concern about. So co-receptors also play a huge role. One more thing, that when HIV enters a cell, becomes reverse transcription occurs, integration occurs, then it forms pro-virus. And once it forms pro-virus, this when it forms a pro-virus, this pro-virus now, first and foremost, forms several copies within the first 3 to 6 weeks. This period, in which it forms its copy, is known as symptomatic stage. Symptomatic stage, or we can also call it as HIV's initial attack. So, symptomatic stage, in which it lasts for 3 to 6 weeks, in which it causes the virus, it causes to produce its copies, as well as it possesses another protein. It possesses two special proteins known as tat and eve. Tet and Eve are two proteins which it produces, and these proteins basically act as inhibitors in regulatory system such that when these proteins act as inhibitors, they tend to inhibit the gene regulation of the host cell's genomic expression, such that when host cells genes are expressed, it acts as inhibitor to decrease the rate of protein synthesis but does not completely inhibit the protein synthesis, so it somehow acts as inhibitors as well. So these proteins, as they act as inhibitors for the protein synthesis, it causes to you know decrease the rate of protein synthesis, as a result of which provirus starts expressing its genes, as a result of which it produces its own proteins, and thus replication is possible to occur. And during this period, several copies are also made. Our body's immune system also fights against those viral copies, but our body's immune system is only able to fight against those viral copies, but not the actual provirus which is present integrated within the host cell genome. So, because there in our body there is no mechanism to kill the T helper cells, because T helper cells are themselves the immune cells which uh, you know help in providing defense against foreign pathogens. So as a result of this, T helper cells cannot have uh, any sort of a defense mechanism to kill them. Right. Likewise, if you are having provirus, now provirus cannot be destroyed because a body doesn't have a mechanism, and T, as it is integrated, it remains within uh, the T helper cell. And after that period, when virus sees that there are some unfavorable circumstances, so it's not a good time for me to um express my abilities. It's not a good time for me. So what it does, it enters into asymptomatic stage. Asymptomatic stage, or in other words, we can also call this stage asymptomatic stage, or asymptomatic stage, or dormant stage, latent stage, in which the virus stays silent for 8 to 10 years, or maybe more or less than that, but there's a general period after which it either produces a few copies or produces no copies, and it stays silent for a much amount of time and inhibits its genes by turning its genes off using the inhibitor. And with the help of this, it causes to produce it causes to find unfavorable circumstances. Now, um, within this period, there are no symptoms visible, so it is on its asymptomatic stage, and it lasts for around 8 to 10 years, or around several years. And one more thing, that here, um, this asymptomatic stage, this asymptomatic stage, dormant stage, has of course, T helper cell has provirus, so this provirus will start to replicate. As it will start to replicate, it will cause uh, T helper cells will replicate during such a long period of time, due to which when they'll replicate, the provirus will also replicate, so their copies will also be made. And now, final period, which is AIDS is the last stage in which the virus is completely spread all over the body, and once it is spread, it finds favorable condition, it then showcases its real properties. It just causes a condition of AIDS, in which several symptoms occur, as well as, example of this is um, several types of opportunistic diseases. Opportunistic diseases are those which are due to weakened immune system within our body, such as pulmonary tuberculosis, as well as fever or cold, or even minor diseases which are caused within our body can seem a more deadlier disease because of the presence of weakened immune system. So opportunistic diseases occur. Opportunistic diseases. Moreover, AIDS also results in the form AIDS also results in opportunistic diseases and special types of cancers, a skin cancer which is known as Kaposi's sarcoma. Kaposi's sarcoma. So these are the things which we are also going to discuss in our next video. That's only for the intro. Hopefully we'll meet again in another one.